So look, in the last class, what I told you that what is the transforming principle? Means the genetic material. What is the genetic material? So in the starting days, the genetic material used to say transforming principle, right? And then after some time, it was considered that transforming principle can be of two types. Means genetic material can be of two types, any of that. So most of the scientists before 1944, before the 1944, most of the scientists, or rather majority of the scientists, used to understand and used to think that this is the protein which is the transforming principle. Means this is the protein which is genetic material. So protein bring down the characteristic of parents to their offspring uh, from one generation to another, and that's what that is the function of protein. But after these, some of the after the experiment of this Frederick Griffith, and after the confirmation of this experiment, so Frederick Griffith did this experiment, then it was confirmed by MacLeod, McCarthy, and Avery, and after that, finally, Michelson and Settle. So they did their experiment, and after these number of experiments in 1944, it was proved that DNA is genetic material, not the protein. And after that, the concept changes that DNA is the genetic material, and it brings down the characteristic from one generation to another generation. So this was the discovery of the genetic material. So in the starting of one of the landmark discovery was transforming principle. So there was a scientist, his name was Frederick Griffith. So the question have been asked in 1926, he did this experiment. What was the question? Actually, the question was that which organism is used for this principle, right? So actually, he used a experimental organism. It was a bacteria. The name of the bacteria was Streptococcus pneumoniae, and this bacteria is responsible for pneumonia in mammals. Rat is also a mammal. So this bacteria, Streptococcus pneumoniae, is responsible for pneumonia in the mammals. And Streptococcus pneumoniae have two kind of this strain. One of the strain is disease causing, another is not disease causing. So what is that? What is the structure of this Streptococcus pneumoniae? This is the cell of Streptococcus pneumoniae. Around the bacteria, there is a layer, slimy layer. This slimy layer dried up in the higher temperature, and it makes a capsule. So capsule may be of two types. One of the bacteria, one kind of or one strain of the bacteria have rough capsule, and another bacteria have the smooth capsule. The bacteria having the rough capsule, they are non-virulent, means they will not spread the disease, they will not cause the pneumonia to mammals, right? And in short, we call them R cell, means the cells which having the rough capsule. Another kind of the species or the strain of the bacteria which have this smooth capsule, they are the virulent. Actually, they causes the disease pneumonia, and due to that, mammal die. In short, we denote them with the S cells. So there are two type of the cells, R cells and S cells. R cells are non-virulent and S cells are virulent. They are disease causing cells, right? Now he used two kind of the cell. So what was the experiment? Come to the experiment. Frederick Griffith first taken the living R cell, rough cells, means non-virulent cell, and he injected these non-virulent cell to the mouse, right? When this non-virulent cells were injected, mouse did not die, and the blood sample was taken. Number of the non-virulent cells were present over there, means they multiplied. After that, what he did, he taken S cells, smooth cells, disease causing, means virulent one. They were injected inside the mouth, and after some time it was seen mouse died, and number of virulent cells were found over there, means the smooth cells are found. Then he did one more experiment. What he did, he killed these S cells, means virulent cells, with the heat. He given a heat to virulent cells, then killed them. After killing them, he injected inside the mouse and found that mouse did not die, because the virulent cell died, and because of the death of the virulent cell, they could not cause the pneumonia to mouse, right? And mouse survived. In the fourth experiment, what he did, he taken these heat killed S cells, means non-virulent, and then non-virulent living cells, and he injected inside mouse, and surprisingly the mouse died. Now this was the very strange. Why the mouse died when two kind of the cells were mixed? Heat killed S cells and these living R cells. In both of the conditions, they are the non-violent. Then after the blood sample, it was found that in the blood sample, the cells which injected were heat killed, smooth cells. But the living S cells, smooth cells, were found in the blood sample. Now, it was very surprising. So, on the basis of this experiment, Frederick Griffith, he concluded that there is some chemical, there is some substance which is transferred from dead smooth cells and transferred to rough cells, and it changed the nature of the capsule of rough cells and converted into smooth cells. He said this is the transforming principle. Transforming principle is a type of the principle which which transferred the characteristic of smooth cells, dead smooth cell, into the R cells, and by this way it converted these rough cell into smooth cells, and it was called transforming principle. Actually, what happened at that time? Actually, from the part of the DNA transferred from S cell to living R cell and converted the characteristic of R cells into smooth cells and caused them virulent. So this was the transforming principle of Frederick Griffith. Tell me, is it clear or not? And if it's clear, you can take the screenshot. That's it. This is the information, and these are the questions for you. Is that clear to all of you? Clear? Heather, clear?
Yes, sir. So how did it get converted to S? How did the living R cells get converted to the living S cells?
Actually, what happened when we mix both kind of the bacteria, plasmid from the smooth cells, it came out from the smooth cells, and it inserted, like it penetrated and got inserted inside the rough cells. And when plasmid reached into protoplasm of the rough cells, it started doing its function, and the cell died, but plasmid did not die. And the plasmid got inside the R cells, and it started producing the slimy layer around the rough cells and made this capsule smooth. And this smooth capsule have a particular kind of the carbohydrate, which is uh, glyco that was glyco sugar, that was the virulent, and it caused the disease. So
Okay, sir.
that's how it happened, right? Basically, the Avery was not aware with the term genetic material, so he used the transforming principle in place of genetic material. Clear, all of you?
Yes, sir.
Okay. Betsy, is that your name? Iram, clear? Iram, are you there? I'm unable to listen you. Okay, so next experiment. Now the next experiment, this experiment was a conformation kind of the experiment, right? And this was the biochemical nature of the transforming principle. Now this experiment tried to prove that what is the biochemical nature, means what is the actual nature of the transforming principle, right? So biochemical nature of the experiment was proved by three scientists, right? As I told you, they did not did any new experiment. Rather, they have carried forward the experiment of Frederick Griffith, right? So there was three scientists, as I told you: Oswald Avery, Oswald Avery. I told you Avery, MacLeod, and McCarthy. There are three scientists. Colin MacLeod, Colin MacLeod, and Maclean McCarthy. So Oswald Ovry, Colin MacLeod, and Maclean McCarthy. There were three scientists. They again try to prove this experiment of Frederick Griffith. So what was the transforming principle? Means whatever the Frederick Griffith discovered, what was that substances? So what he did, they all these three scientists, they taken the heat killed S cell, smooth cells. After taking this heat killed smooth cell, they removed from this suspension, they removed lipid and sugar. Two chemicals were removed with the help of enzyme. Now, what is the left with this chemical? The three things left with this chemical, right? What is left? This suspension contained three things: protein plus DNA and plus RNA. So three things left. So remove the lipid and sugars from the solution of heat killed S cells. Now the protein, RNA, and DNA remains, right? In first experiment, he added a proteinase, right? The treated solution with the enzyme proteinase. With the help of treated solution with the enzyme to destroy the protein, RNA, or DNA, right? So then he added a proteinase and destroy the protein. Now what is the left with there? That is DNA and RNA is left with there. Now what he did, he added the R cell, rough cells, and he like they saw over there that the R cell converted into S cell, right? Means the rough cell transformed into smooth cells. It means either DNA or RNA is transforming principle, not the protein, because protein is not present. In the next of experiment, again, he taken the solution and he removed two things. He removed the protein, added the proteinase that destroyed the protein, added the RNase. RNase is an enzyme that destroy the RNA, digested RNA. He destroyed the RNA, destroyed the protein. Now the only thing left, that is the DNA. And he again added the rough cells and found that smooth cells appear. It means the DNA is the transforming principle, because in this solution only DNA is present and the rough cell converted into smooth cells. That's how he proved. Again, in the last experiment to confirm, in this solution, lipid is not there, sugar is not there, protein is not there, RNA is not there. Now he added the DNAs, right? When he added the DNAs, this enzyme destroyed the DNA, digested the DNA. Now the R cells do not convert into smooth cells. With the help of this experiment, Oswald, MacLeod, and McCarthy, they proved that this is the DNA which is the genetic material, not the any other substance. So he said that they said that the transforming principle of the Frederick Griffith is DNA, not the protein. Please note it down. Every MacLeod and McCarthy, every MacLeod and McCarthy proved that the transforming principle is DNA, neither protein nor RNA. Please, if you want, take the screenshot. Biochemical nature of transforming principle. Taken?
Yes, sir.
Okay.
Sir, in this experiment, the
Yes.
interaction will happen between R cells and S cells, right? So how
Right.
does it matter if the solution has DNA, RNA, or protein, anything at all?
No, not at all. Not at all. Only in presence of DNA, the R cell convert into S cells. They want to prove which chemical converted these rough cell into smooth cells. So possibility where it may be protein, it may be RNA, or it may be DNA. So out of these three possibility, they did the experiment and checked which of the following is responsible. Finally, with the help of their experiment, they find out that this is the DNA which is responsible for transforming rough cells into smooth.
Sir, how does DNA take this process out?
The
Like,
same
how
thing
does it help
that
it?
actually what happens when the temperature difference takes place. The wall of bacteria, they become porous, right? Plasmid come out of the bacteria. One thing. Second important thing, when the bacteria come in the contact, there is a process of conjugation. With the help of conjugation, what bacteria do, they make the link with another bacteria. Two bacteria come in contact with each other, right? When it is also called that sexual reproduction in bacteria. Two bacteria come in contact with each other, right? And they make a conjugation tube or conjugation bridge. If you will put this topic in internet, you will get it, right? Conjugate, that is out of actually your syllabus, but I'm telling you how it takes place.
Yes, sir. Yes.
They make a conjugation bridge. When they make a conjugation bridge, a part of the DNA transfer from one bacteria to another bacteria. And this process, conjugation process, is also known as sexual reproduction in bacteria, right? And the part of DNA which is transferred from one bacteria to another, the characteristic it bear, the part which bear that characteristic, that is transferred to another bacteria. The same thing happens over here. Got it?
Mm-hmm. Okay, sir.
That's how it transfers. With the process of conjugation, actually, the female bacteria get converted into male bacteria.
Yes, sir. So this is the conjugation is the process where F plasmid is getting transferred, right?
Exactly, exactly. Now you got my point. You got my point. Yes. But actually, it has not been covered in the NCERT. It's not part of that. So that's why I was not discussing.
Okay.
You will find this like I'll brief you this one. The whenever we transform the bacteria, whenever we transform the bacteria, when we insert the plasmid, right? Suppose that I have a in biotechnology part, I will tell you. Whenever we have the plasmid, we change the temperature difference, and with the help of temperature difference, it absorb the plasmid itself without any conjugation process. That is also a way to take the plasmid. Just you have to uh, differentiate the temperature, high, low, high, low, high, low. That's it. So now this is the experiment, which Hershey Chase experiment, which finally proved that DNA is the genetic material. If you this question have been asked, so I'll tell you this question have been asked, and if you will go through with the previous year question paper, you will find that which of the following experiment prove that DNA is genetic material. Then on that case, you have not to tick every MacLeod and McAfee. You have to tick the this experiment. Uh, this experiment of this one, Hershey and Chase experiment. Then Hershey and Chase experiment explains that DNA, and it finally proved that DNA is the genetic material, not the protein, right? So come here. Here we have used two radioactive substances. First, come to the structure of the virus. So what he taken in this experiment, he taken a bacteria and the virus, which is so which bacteria? He taken the bacteria of Escherichia coli, right? The bacteria which was taken in this experiment, that is E. coli, right? And then the virus which is taken, that is the bacteriophage virus. Bacteriophage virus, sometimes it is also known as the phage only, right? Bacteriophage virus is made up of two things. The outer cover is known as capsule, right? This outer cover, which is known as capsule. This capsule or outer cover is made up of protein that is called capsid protein. Protein contain the sulfur, right? Sulfur. So here to radio label the protein, we have taken sulfur 35 S35, right? In the same way, inside the core, there is genetic material which is the DNA. We have radio labeled the DNA here with the phosphate 32, right? So that's how we have radio labeled. In experiment, first, one more important thing which I want to share with you. Whenever a virus infects the bacteria, the outer covering, this protein shell or protein covering of the bacteria, it remains outside. It do not goes inside the body of the virus. Actually, the capsule part which remains outside, that is known as the host. And the bacteria whom it infect, that is called host, right? Look, so in first of experiment, what we did, the normal protein do not have sulfur 35, but we have radio labeled the virus with the help of sulfur 35. Because the why the sulfur? Because sulfur is found only in protein, not in DNA. And the same thing, phosphate is found only in DNA, not in protein, right? So these are the two things which is the different in these two chemicals. That these two biochemical. That's why we have taken sulfur and phosphate. If you ask me why you have taken only, why Harsh and Chase taken only phosphate and sulfur, then this is the explanation. Actually, the sulfur is found only in protein. It makes sulfur bridge in protein, right? And the phosphate is found in the DNA. It make the phosphate sugar backbone, right, of the virus, of the DNA. So here what we did, we radio labeled the protein with the help of Sertal 35. Now we have taken this virus and we infected a new bacteria. Virus infected the bacteria, outer cover fell down. Now this host part remain outside of the bacteria. Now it was found that there is no radioactivity. It means the protein is not genetic material, right? Because radioactivity was not found inside the cell. Again, in next experiment, this time we have radio labeled the DNA with the phosphate 32, right? After radio labeling, again we infected the fresh bacteria. Again, the ghost is outside of the cell. After multiplication, we find that radioactivity is present inside the bacteria. It means it is proved that this is the DNA, which is genetic material, not the protein. That's it. So that's how the Hershey and Chase proved that the DNA is genetic material, not the protein. Is that clear?
Yes, sir.
Okay. So please take the screenshot and remember what was the radio labeled. These both were the radio labeled, right? Now come to the next experiment. I'm not getting the response of Betsy and Iram. Iram, is it okay?
Yes, sir.
Very good. What about you, Betsy?
Yes, sir, it's okay.
So this is your name, or it's your just
Yeah, it's correct.
Okay.
Betsy.
okay? Fine. So next experiment, that DNA is semi-conservative. You will get the, when you will solve the previous year question paper, so you will get the questions from there, and I think somebody from my team, management team, they must have shared with you, right, that previous year question bank. Do you have?
Yes, sir.
Very good. So Arihant is good one because it have explanation as well. Otherwise, in the rest of their rest of the publication, there is no explanation, right? So keep on solving the previous year question paper, and by this way, you will get the confidence that you are in the right track or not, right? And you will see, you will get the question what we are discussing in the class. So, what is the semi-conservative model? Let me first explain you what exactly this model is. Semi-conservative. There are some people who are fully conservative, right? They don't want any kind of a change. Semi-conservative people are those which keep some principle and incorporate some new thing, right? Okay, they live with some of the traditional old principles, but along with them, they incorporate some new thing as well, which is required, right? That is called semi-conservative kind of people, right? So in conservative, there is no change. So in the same thing happened with the DNA. So DNA is semi-conservative. So suppose that we know that there is a property of genetic material, that genetic material must be capable of duplicating itself, right? So this is the parental DNA. When replication of DNA takes place, what happened? This DNA open into two forms. This is called replication path, right? Right. This black color structure shows that this is the parental strand, right? This is the parental strand, and the red color, this is the new one, right? By this way, the replication reaches at the last end, and we can see this DNA, it have 50% of the old one, right? Means parental, and 50% the DNA of new one, right? 50% new DNA, right? So there is a this the base is parental, and the new DNA means parental strand, and the new DNA is formed on that strand, right? This is called semi-conservative. This model have been proved by two scientists, and what they did and what they use in their experiment, basically they taken the NH4Cl, right? NH4Cl, ammonium chloride, right? And this experiment, it was done by the two scientists, and which bacteria? That is also very important. Let me write it down so that when you take this screenshot. This experiment was also done on the Escherichia coli, E. coli, because these are the small experiment, and sometimes you get confused when it comes to your examination. So even this experiment, semi-conservative experiment, was done on E. coli, and again the two scientists did this experiment, and in 1958, two scientists. The first scientist is Matthew Michelson. So Michelson and Franklin Stahl. So you can remember Michelson and Stahl did this experiment. Michelson and Stahl did this experiment in 1958, right? And they grew the bacteria in the medium that is NH4Cl. Even this question have been asked. Remember NH4Cl. Now here, this two kind of the nitrogen are found. Means two type of the isotope of the nitrogens are found. Remember, N15 is not radioactive. N15 it do not have any kind of the radioactivity, means it is not radioactive. In the previous experiment, that sulfur and phosphate were radioactive, radio labeled. They have the radioactivity. Here, these are the lighter isotope and heavy isotope. So there are two kind of the nitrogen, N14 and N15. N14 is light and N15 is heavy one, right? Now, first, what they did, they taken a medium where they grow the E. coli, right? And that medium having N15, right? After that, the multiple time of when bacteria multiplied, the bacterial DNA or DNA is made up of nitrogen 15. And everywhere the density was with the nitrogen 15, means heavy density. Now the bacteria is allowed to grow on the normal medium that contain N14. When bacteria grown in normal medium, it was found that the density was between N15 and N14. It means the 50% strand was made up of nitrogen 15 and 50% strand was made up of nitrogen 14. Again, so now you can see here the density is 50-50. So 50% parental strand and 50% new strand. Again, when it was grown, it was seen that the 25%, right here it is 50-50, means 25% density of this one, nitrogen 15, and then 25% of nitrogen 14. Again here 50% the density of N14, right? So every time now the density of N14 is increasing. By this experiment, they proved that DNA, the replication of the DNA is a semi-conservative and follow the semi-conservative pattern, and this is called semi-conservative model of replication. Tell me, guys, is that clear? And if you have any question, please ask. Clear, Heather? Betsy
Yes,
and
sir.
Iram?
Yes, sir.
Now, I have a question. When they separated the DNA, extracted the DNA, and get the density of DNA, to get the density of these DNA, they used a special type of the equipment. What is the name of that equipment? With the help of centrifugation, they got the density of N14 and N15. So what is that equipment? This is the question. It has been asked. That's why I am asking you. It is directly from the NCERT. Please note it down. To get the density, NH4Cl, this question I have been asked, and again this question I have been asked. To get the density of DNA, they used to get the density of DNA, they used cesium chloride, CSCl, cesium chloride density gradient, right? Density gradient, centrifugation, centrifugation technique, right? So they're given they with the help of CSCL centrifugate, they got the density of DNA, right? It is given in NCERT. What is the specialty with the cesium? Cesium is very special kind of the metal. What is the specialty with the cesium? Anybody of you? No? Cesium? You must have uh, studied in your this inorganic part of the chemistry. No? Cesium? Cesium is the poisonous metal. If anybody eats cesium, he will die. First thing. This is the metal which is used in atomic clocks. What is the purpose of atomic clock? Where we use the atomic clock?
Space.
Yes, right. Space. The time you get in your GPS system, the time you get when you drive your car, the time you get in GPS system, that time is decided by that space clock, right? It is they use this atomic clock. Sorry, atomic clocks. Atomic clocks are placed in the satellites only, right? And there, for making the atomic clock, we use the cesium. It's very precious metal. It's poisonous too. This was something strand formation. Now come to another experiment. DNA replication. What they will ask you if from the DNA replication? They will ask you directly from the what enzymes, right? Okay. So how does the DNA replication takes place? You have to take the screenshot of each and every part, right? Very important. If you'll go through with this diagram only, this PPT only, you will get like every question which is being asked in your examination. So first, we know that when replication takes place in semi-conservative model, I told you that when the replication takes place, during the time of the replication, a DNA open, right? So opening of fork takes place, means the DNA open, unwind the DNA, unwinding of DNA takes place, and due to unwinding of DNA, two parental strand get formed, or formation of two parental strand takes place. And when formation of two parental strand takes place, on that time, new strand formation takes place on the parental strand. So parental strand, they behave like template one, and formation of new strand takes place on those templates. So now we start from here. And in case if you have in case if you have any doubt, please feel free to ask me, right? Don't hesitate, because you won't get this time again, right? This is the time when you can ask me, right? Look, first opening of DNA, so which work like Caesar, which cut down the DNA from the mid, which cut that ladder from the mid, right? There is an enzyme, the name of the enzyme is helicase. So I have written over here what is the function of helicase. Unwinding of strand, it cut down the DNA from mid, unwinding of the strand takes place, and formation of fork takes place. With the help of these helicase enzyme, formation of fork takes place and unwinding of DNA takes place, right? So first function was of helicase. Then the second enzyme is topoisomerase. When fork open, then strain get created, formation of a force, opposite force develop at here at this place, and this is a strain force, right? This is strain because DNA is twisted. When unwinding of DNA, whenever you open that wire, black and red color wire, you must have seen, if you start opening of that wire, what will happen? The strain get created or strain develop at the neck of that wire. The same thing happen over here. Whenever we open the DNA, because DNA is a helix-like structure, a strain force develop at the neck of this fork. Now there is an enzyme which will remove this strain. Otherwise, due to the strain, formation of the fork will not go ahead. Fork cannot move ahead. So what happens actually? There is an enzyme. The name of that enzyme is topoisomerase. Topoisomerase, what do? It remove the strain. How does it remove the strain? It make a cut, then a strain removes, and again rejoin DNA. So that's what continuously it keep on doing. So by this way, topoisomerase remove the strain by cutting and rejoining the strand. So that is the function of 2 points. Now the fork have opened, right? This single strand here, single strand here. Now what can happen that this single strand can rewind. Again, the formation of they can restrict. The formation of bond can take place. So these single strand do not rejoin or restrict. So prevent them. This question have been asked, right? Which of the following is non-protein? Which of the following is non-enzymatic protein? Answer was SSB. Question have been asked AIMS and NEET both exams. Look. Now, if these single strand a protein bind over here, which do not allow them to rejoin. The name of the protein is SSB, single strand binding protein. It is non-enzymatic protein. This question have been asked in medical examination. So this SSB is it is protein, but it is not enzyme. Or you can say which is the non-enzymatic thing which participate in the replication of the DNA. Basically, this is the SSB protein, which is the non-enzymatic thing, and it is taking part in the replication of the DNA, right? It is not enzymatic protein, and it do not allow single strand to rewind. I have written over here, right? Now the fork opened, right? And there is enzyme. Now you can take the screenshot. Please take the screenshot. Then I'll go ahead. Look. Now the main function is of the fourth enzyme which work over here, that is DNA polymerase. Now the functionality of DNA polymerase can only work in the 5 to 3' end, right? It will only move, it will go ahead, means polymerization reaction can be done only 5 to 3' end, right? And continuous the polymerization reaction. It make the DNA on older strand into 5 to 3 direction. Now look. Here, this is the lower one, this is the parental strand. So parental, this is 3' end, this is 5' end. Now DNA polymerase is smoothly making the new strand on 5 to 3' end. It is continuously making, right? That's why it is called leading strand, right? It is called leading strand or continuous strand, right? This strand. Okay. So DNA polymerase is making the new DNA in 5 to 3' end direction, right? When the DNA formation takes place, first enzyme work. The name of the enzyme is primase. Primase bring RNA primer, right? First RNA primer will join. Here the RNA primer will join. Then the formation of DNA strand takes place, right? But in starting, there will be the small fragment of the RNA, which is called RNA primer. So this is called leading strand. But in opposite strand, so the fork opening takes place in this direction. Fork opening takes place in this direction, where 5 to 3, and again the DNA polymerase is also moving in the direction of 5 to 3 direction in this strand. But in case of this strand, this is something opposite, right? Now the DNA polymerase, now this is the 5' end and this is the 3' end, right? Now what will happen here? The movement of DNA polymerase in this direction, right? So now the DNA polymerase is moving in the direction of opening of fork. So first it will make a small fragment, then it will come back. Again make a small fragment, then it will come back. By this way, in above this strand, the DNA polymerase have to move in opposite direction, but it will make a small fragment, then it will come back. Again make a small fragment. Joining the RNA primer, then it will move from here to there, make a small fragment. By this way, in above strand, in opposite strand, other than the strand which is other than the leading strand, there is a strand where the formation of a small fragment of DNA takes place. These small fragments were discovered by a scientist. On the respect of that scientist, we name this fragment Okazaki fragment. This question have been asked, right? You have you can get this question. So Okazaki fragment, you have you'll get this question, right? You can get this question. So in above strand, the formation of new DNA strand is not a continuous. That's why we call it discontinuous strand, and it is also called lagging strand, right? So first function of DNA polymerase to make new strand on the older strand. But this is not only the function of DNA polymerase, because in between there are some fragment of the RNA which has been added by RNA primer, right? Now second function of DNA polymerase to proofreading. In proofreading, it go again back, so it will come back, it will remove RNA, and after removing RNA, formation of DNA takes place. So this is called, or if there is something error, like it have done some mistake or some error, then proofreading also the function of DNA polymerase. So that's how the formation of new strand takes place on the older strand, and that's how the replication takes place. Tell me, guys, is it clear or not? And please ask me if you have any question. All of you, please tell me. Betsy, Iram, Heather.
Yes, sir.
Yes, sir.
Very good. Betsy, what's about you?
Yes, sir.
Okay, this is self-explanatory diagram. So I request you to please take the screenshot of this one. And the function of DNA polymerase. So polymerization in 5 to 3 direction, that you have to remember, 5 to 3 direction. It also perform the proofreading. What is the function of primase? Make the RNA primer. Now, what is the function of ligase? So when it fill the gap, so that gap should be added with the help of glue. And there is an enzyme named ligase. That ligase add these separated part with the help of that glue. This Okazaki fragment finally added by help of a glue, and this is also called molecular glue, and the name of that enzyme is ligase. One very important information that have also been asked multiple times. In prokaryote, helicase and topoisomerase enzymes are not found. Right? In prokaryote, if you want, you can note it down. In prokaryote, helicase means which open the helix, topoisomerase which remove the strain. So helicase and topoisomerase are not found, right? So in place of helicase and topoisomerase, the enzyme which work, that is the gyrase. So gyrase enzyme. So in prokaryote, in place of the topoisomerase and helicase, gyrase work, right? Okay. DNA polymerase third. So there are type of the DNA polymerase. DNA polymerase third, it work in prokaryotic polymerization, means it function in prokaryotes, and DNA polymerase alpha and mod, or DNA polymerase 1 and DNA polymerase first and second in work in eukaryotes. These were questions. They all were questions. Hope you must have taken the screenshot.
You can just scroll up to the diagram once.
Okay.
So in the leading strand, the DNA polymerase works on the new strand, and in the lagging strand, DNA polymerase works on the original DNA strand.
In both of this one, original DNA strand, it is also original. This one, this lower one, this one, right? This one, right? So this is this green color structure, lower one structure. This is the original strand. Here also original strand, right? This is the new strand, and this is the new strand, right?
But sir, in the lagging strand, the original strand is in 3' to 5' direction.
Look here. What is happening? This is the 5 to 3 prime. This is this one, right? You can see. This is the 5. This is the 5 to 3 prime direction. This one, the direction is 5 to 3 prime, right? This is the older strand, right? This is the parental strand.
Yes,
So
sir.
this is 3 prime. This is 5 prime. Got it?
Yes, sir.
And this is the 5 prime. This is 3 prime. So here in this one, this direction. This is the 5 to 3 prime direction. In new strand, DNA polymerase is going on like this way. But in here, this is the 3 to 5 prime. This is the 5 to 3 prime direction. This one, this direction. So what it will do? First, it will make a strand from here, and then it have to come back. Then it will go back. Then it have to come back, because in the lower one, the direction of the fork, opening of fork, and direction of DNA polymerase is same. But in the above one, you can see the direction of the fork formation is this one, and direction of the DNA polymerase is this one. So what it will do? It will go first, it complete, then will come back, then complete, then will come back, then complete, then will come back, right? So that's how it have to do. So when it do this practice, what happens? The formation of a small fragment takes place.
Okay, sir.
And those fragments are called Okazaki. Okay.
Okay, sir.
So here in lower one, there will be only one RNA primer. But in above one, every time it make fragment, it have to add on the RNA prime. Got
Okay.
it? Then the proofreading will be done, right? Transcription. What is the transcription? Formation of RNA on DNA strand. So can anybody tell me what is the basic difference between the transcription and replication? Any one of you? What is the basic difference? Very basic difference. Main difference. What is the basic difference in transcription and replication? Look.
The
What is the basic?
transcription makes RNA from DNA.
Right.
The replication makes DNA from DNA.
Okay. What is the difference between the process? That's what I am asking. What is the main difference?
Sir,
Look. Very first thing that when formation of DNA takes place, the complete fork have to open. Right? DNA will be completely open into two parts and the new strand will be formed. Here, it's not like that. A small part of the DNA will open, formation of RNA will take place, and again that will remain. First, very important. So, fork formation have not to take place here in RNA formation, right? Means transcription. Second thing, in replication, formation of new strand takes place at the both of the strand, leading and lagging, both of the strand. Both of the parental strand will work at template. Here in RNA formation, means transcription, formation of RNA takes place only on one strand, on template strand, not in second strand. On the second strand, in the other strand, formation of RNA do not take place. That is a very basic difference. Now come to the what is the transcription. So simply I told you that the transcription takes place. So formation of mRNA, messenger RNA, on the DNA is called transcription, right? Messenger RNA is also known as hnRNA, heterogeneous RNA, because the length of messenger RNA is not defined. It depends on the length of the gene. If the gene is 1000 or 1 lakh nucleotide longer, then the RNA will be 1 lakh nucleotide longer. If the gene is 100 nitrogenous base longer, then the RNA will also be 100 nitrogenous base longer. So the length of the RNA is not defined, right? They are of different length. That's why they are called hnRNA or heterogeneous RNA, right? That's why messenger RNA normally is also known as heterogeneous RNA. You can see over here. I have written over here, right? Okay. So how does it take place? Look. First here, you can see the local unwinding taken place. Okay. I want your 100% focus over here. This is a bit brainstorming kind of thing. One more important thing I want to tell you that the movement of DNA polymerase or polymerization reaction of DNA polymerase is 5 to 3' end, and the same thing with the RNA polymerase. It will also polymerize in 5 to 3' end. Okay. Now opening or unwinding of strand taken place. Now what will happen? This RNA polymerase will scan DNA and it will try to find out a particular sequence. That sequence is called recognition sequence. So there is a promoter gene. This promoter gene have from where the RNA formation will start. Before the RNA formation start, at the starting of RNA formation, or starting of the transcription, in that strand, DNA strand, there will be a recognition sequence. That will be promoter gene. Promoter gene have a recognition sequence. This RNA polymerase will scan complete DNA and it will find out the recognition sequence. Whenever it will find out the recognition sequence, it will land over here. Now in RNA polymerase, which part recognizes the recognition sequence? In RNA polymerase enzyme, there is a protein named as sigma subunit. This sigma subunit of the RNA polymerase recognizes the recognition sequence and it land over here. After the landing, it starts the formation of RNA on the DNA template. It keep on the forming. So this is the gene, and the starting of gene, there is a recognition promoter gene. Promoter gene have recognition sequence, and you can see this is the 3' end to 5' end in parental DNA strand, and the RNA strand you can see this is 5 to 3'. So as I have told you, the RNA formation will take place 5 to 3'. Now, at the end of the gene, there is a terminator sequence, which will terminate the process of RNA formation. Terminator sequence is recognized by Rho protein. It is also found in the RNA polymerase. If you want, you can note it down, right? I have written, by the way, I have written over here, this one, right? The sigma subunit of the RNA, polymerase recognizes the recognition sequence at the promoter site, and Rho factor of the RNA polymerase recognizes the terminator sequence, right? So at the terminator sequence, it will be recognized by the Rho factor. RNA polymerase will dissociate, then this RNA will come out. Now, one important thing, where the RNA formation will take place, in which the strand of DNA on which the RNA formation will take place, that is called template strand, and this template strand is also known as an antisense strand. The opposite strand is called non-template, because this is not working as a template. Along with this is called sense strand. So this is very strange. Where the DNA formation is RNA formation is taking place, that DNA sequence is called antisense. Why? Look, why we call this one sense strand? Because you can see, look, read the RNA sequence. In only the difference is U and T. You know that there is a difference between the RNA and DNA. Uracil and thymine. Look, UAC, TAC, GCC, GCC, AG, AG, AG. So this RNA is copy of this one. RNA is true copy of this one. That's why this strand is called sense strand, right? RNA is not copy of this one. It is opposite of this one. RNA is the opposite of this one. That's why this is called antisense strand. Did you got this point or not? Tell me.
So like helicase was used to open the DNA in replication. What is used to open the DNA over here?
Here the RNA polymerase will perform all the function. It will open the, it will recognize the this one recognition site, then it will unwind the DNA locally. So all the function will be performed by the RNA polymerase. There is no helicase involved, because it have not to make any fork kind of the thing. That it is just local unwinding, right?
Okay.
DNA unwind. Look, everything is written over. DNA unwind locally. RNA senses, then DNA again rewind after RNA senses. RNA polymerase scan and search for the recognition sequence. RNA polymerase dissociate the DNA at the terminator sequence. Sigma subunit of the RNA polymerase recognizes the recognition sequence at the promoter site. Rho factor of the RNA polymerase recognizes the terminator sequence and RNA dissociate. Please take the screenshot. Taken the screenshot?
Yes, sir.
Now next one. Maturation of DNA. Again, spliceosome have been asked as a question in medical examination. So, whenever formation of RNA takes place on DNA strand, so some part is of gene and some part is of non-gene. Means some part is coding part, some part is not coding part. So there are some coding part. These are called exons. Means they are the real copy of the gene, right? They are called coding part, right? So they are called exon. And there are some useless part, they are non-coding part, they are called introns. So what we need to do in any RNA, in RNA, there are a number of the stretch which are the non-usable. They are called introns. So we have to remove intron over here. That is called maturation of RNA. Then what happened? There is a splicing protein. The splicing protein join at the part of the intron part after the DNA, after the RNA transcription. They make a hairpin loop-like structure. These structures are called spliceosomes. These spliceosomes are split or cut out from these RNA part with the help of this splicing protein. These introns are spliced off, and again these exons reunite. Right, so these exons reunite, and that's how the maturation of RNA takes place. You have to remember what is the splicing protein and splicing. It was simple. Clear? This is very important. So spliceosome have been asked. Again, the maturation of DNA. So that splicing part if had taken place inside. Now, if this naked RNA will come out of the nucleus, this is nucleus. And now, what is the purpose of RNA? RNA take out the information of DNA. It take out the information of DNA. And after getting the information, it have to make the protein on the instruction of DNA. From DNA take the gene copy. Then it come out of the nucleus with the help of, there are some pore, these are called nuclear pore, come out of in protoplasm. In cytoplasm, it have the ribosomes. It goes to ribosomes. And as per the instruction of DNA, they make the polypeptide chain or protein. That is the purpose of messenger RNA. But if the messenger RNA will come out like this way, it will be eaten out or destroyed by enzymes which are called hydrolytic enzymes. These hydrolytic enzymes, they are found in the protoplasm, right? So now we have to protect the RNA, mRNA from the hydrolytic enzyme. What happened? This is the coding region, and at the start there are some uncoding region, and at the last there are some uncoding region. So at the start site means at the this one 5' end, their formation of a cap take place. That sequence is called methylated guanosine. So at the 5' end, methylated guanosine add on over here, and it make a 5' cap, right? It make a cap which protect RNA polymerase from hydrolytic enzyme. At the 3' end, adenine join, number of adenine join, and they make a polyadenine tail. So when the RNA comes out, before coming out, formation of methylated cap take place at 5' end, and formation of a poly A tail take place at 3' end, so that it could be protected from the hydrolytic enzyme when it comes out in the protoplasm or cytoplasm. Tell me, guys, is it clear or not?
Clear, sir.
Okay. So you have to remember methylated cap at the 5' end and poly A tail in the 3' end, and why? Because of hydrolytic enzyme. One more important thing which I want to first ask from you: what are the monocistronic gene? What are the polycistronic gene? Polycistronic gene are found in prokaryote, and monocistronic gene are found in eukaryote. Tell me, who will tell me? Hiram, Betsy, Heather, please tell me.
Sir, monocistronic genes are present in the RNAs which can only code for one protein,
Very good, very good. And polycistronic?
and they can code for multiple proteins.
Very good, very good. So in the same way, we can say monocistronic gene, they have the information for one gene, and they make only one type of the polypeptide chain. In polycistronic gene, the RNA have the copy of multiple genes. It make multiple kind of the polypeptide chain. Very good. So you have to remember monocystonic are found in eukaryote and polycystonic found in prokaryote. The structure of tRNA. This is called clover leaf model. This is the structure of tRNA. It is called clover leaf model, and this model was given by R.H. Hawley. He was a scientist, R.H. Hawley, and 1965 he said that tRNA look like clover leaf. And in 1965, for this model, clover leaf model, he got the Nobel Prize for the same. R.H. Hawley, you have to remember. This is the structure of tRNA. What is the purpose of tRNA? tRNA is a like person which bring which pick amino acid. One tRNA can pick only one amino acid. Whenever RNA goes to ribosome, ribosome order to tRNA. tRNA bring amino acid from protoplasm, cytoplasm, and give to ribosome. That is the function of tRNA. tRNA always pick the amino acid at its arms of the 3' end. That you have to remember. What was the question? Which functional group is found at the 3' end of the tRNA? That was the question. At the 3' end, and what is the sequence at the 3' end? At the 3' end of the tRNA, the sequence will be CCA. Always there will be sequence of CCA. This was the question have been asked in medical exams. And what is the functional group? Hydroxyl functional group is always attached at the 3' end of the tRNA. And this hydroxyl will make bond with the carboxylic group, COOH group of amino acid, right? So you have to remember the hydroxyl group is found at the 3' end, and the sequence is CCA. This is the structure of tRNA. From this 3' end, it picked the amino acid, and then there are 3 more arms. There are 3 more loop. 1, 2, 3. First loop is called T-Cy-C loop. The question I have been asked, what is the function of T-Cy-C loop? With the help of T-Cy-C loop, it make the attachment, or it attaches with the ribosome. It is called ribosomal site, because with the help of this arm, it attaches with the ribosome. The question I have been asked. T-Cy-C loop is ribosomal site. Again, this loop, this anticodon loop, is actually this loop make the bonding with the temporary bonding with the which one? mRNA. Because mRNA have messenger RNA have a codon, and opposite of codon is anticodon, which is found in tRNA. Anticodons are found in the anti-loop part of the tRNA, right? This is called DHU loop. DHU loop is called enzyme site, right? And actually, the enzyme comes, and the enzyme bind at this site, right? So this is called enzymatic site. Please take the screenshot and tell me.
What was the function of this tRNA?
Which one?
So what was the function of this tRNA?
Function of tRNA. Function of tRNA is to bring the amino acid from the cytoplasm to this one, ribosome. It bring 1 by 1 amino acid. One tRNA bring out 1 amino acid only.
Okay, sir.
That is the function. Is that clear? Betsy and Iram?
Yes, sir.
Very good. Very good. You all are very bright kind of the child. If you are really getting, then your grasping power is good one. Look, you will get this question as well, right? Structure. Again, I will repeat this this thing in the. I'm telling you right now. Again, I will repeat this thing in the cell biology part as well, right? But you will get you will get the number of the question from this concept. That is the structure of. Just I want to tell the structure of ribosomal RNA. That's why I have explained this diagram. So this is the schematic diagram of ribosomes, right? So ribosome, normally this is the 70S ribosome. So ribosomes are of 2 types, 70S and 80S. 70S ribosomes are found in Prokaryote and ATS ribosomes are found in eukaryote. Ribosomes are made up of two subunits, larger subunit and a smaller subunit, right? So, as I have told you that ATS ribosomes are found in eukaryote and 70S ribosomes are found prokaryote. What is S stand for? S is basically Swedberg. Swedberg was a scientist who designed a machine to get the size of ribosome and another biomolecules. So after the Swedberg, we given the unit name of the unit Swedberg. Swedberg is neither a length nor a mass nor a weight, right? So what is Swedberg? Swedberg is just a centrifugation constant, right? So every ribosome is made up of two units, larger subunit, smaller subunit. A smaller subunit in prokaryote is of 30S, in eukaryote of 40S. Larger subunit in prokaryote of 50S and eukaryote of 60S. Again, this larger subunit is made up of three things: 5S RNA, 23S RNA, and 34 kind of the protein. 5S RNA are found in prokaryote, but in place of eukaryote, they are called 5.8 sRNA is found, right? In place of 5S RNA. So 5S RNA is found in prokaryotic ribosome, and eukaryotic 5.8 is found. Here, 23S RNA is found in ribosomal RNA is found in prokaryote, but in eukaryote 28S RNA is found, right? 34 kind of the protein is found in both of them. Again, in small subunit, 16S RNA is found in the prokaryote, 80S RNA is found in eukaryote. Here is 21 kind of the protein, right? So this is the structure of the ribosomes in prokaryote and eukaryote. Is that clear or not? Please tell me. Look, as Swedberg is centrifugation constant. That's why this 50 and 30 is 80, but it is given 70. Right? 60 and 40 is 100, but it is given 80. Take the screenshot and ask me if you have any question.
So what is the SN and scRNA?
Actually, SN means small nuclear RNA. They are very small. They are a type of RNA I have described over here. They are found in nucleus actually. snRNA. And SC means cytoplasmic RNA. So they are very small RNA. They are some of the RNA do not have any kind of the function. They work like enzyme only, right? Basically, the 20 type of the enzymes are found. 20 type of the RNA have been discovered till date. Out of them, we study only 3: mRNA, tRNA, and RNA. Clear, guys? You will get the question. And when you will do the PYQ, you will get the question from here. So genetic code, is that clear to you or not? Anyone of you? Any question? Genetic code, I think it must be clear to you.
Yeah, it's clear, sir.
Okay, yeah, simple. The just triplet of means 3 nitrogenous bases. The set of 3 nitrogenous bases is called triplet nucleotide. They are called the codon, and every codon code for one amino acid. One important thing, and the property of this one, codon, genetic code. You must remember there are 64 codon. Out of them 64, there is a one initiation codon. So at the formation of polypeptide chain at the initiation, there will be AUG code for methionine, right? There are 3 codon which do not code for any amino acid, and whenever these codon will come, the protein synthesis will stop. They are UAA, UAG, and UGA. They are called stop codon or nonsense codon, right? They are called stop codon because they stop the formation of amino acid polypeptide chain, and they are called nonsense because they do not code for any amino acid. They do not make any sense. And rest the property that code are degenerative, they are universal, right? You have to remember there is no comma between the codes, right? You have to remember the property and you must be aware with that. Okay, so codon, it's easy. Now the very, very important process, translation process. Look, translation I will tell you by my own way. First, replication takes place. Means formation of a DNA from another DNA. Right? A new DNA is formed from the older one. Replication. After replication, transcription takes place. Formation of RNA from DNA. After transcription, translation takes place. Formation of protein from RNA. And this complete process is called central dogma. Right? So what is the central dogma? Nothing but central dogma is from DNA, formation of DNA, formation of RNA from DNA. And the process is called transcription. From RNA, formation of polypeptide chain takes place. That is called translation. And this polypeptide chain is further, you can call it protein as well. Sometime formation of DNA takes place from RNA, exceptional. Where does it take place and what we call it?
Reverse.
RNA transcriptase by the enzyme.
Yes, yes, Bestie, you're telling me. Tell me.
RNA transcriptase by the enzyme.
Very good, very good. It takes place in viruses, and the process is called reverse transcription. Reverse transcription. But this process only takes place in retroviruses, like HIV virus, that is a retrovirus. TMV, tobacco mosaic virus, that is a retrovirus, right? So reverse transcription also takes place, and the name of the enzyme is reverse transcriptase, right? Very good. This process of formation of DNA to RNA to polypeptide chain, this is called central dogma. Central dogma. Now, come to this part. What is the translation part? Look, how does the translation take place? Let me tell you a simple story. With the help of this story, I will tell you what is the translation process. Look, suppose that right now I'm in front of you and I'm sitting in this studio, right? This studio room have just a single door, right? And I'm teaching you, I can't go out of this room, right? So, but I need a set of books, that is a 10 books, a set of 10 books. I need that set of 10 books. Right? Set of 10 book consider is as a protein, and every book is amino acid, right? So I need a set of 10 book, means I need a protein. And every book is amino acid, means protein is made up of number of amino acid, polypeptide. Who am I? I am a DNA, right? I am a DNA, and this room or this studio is what? Nucleus. DNA cannot go out of the nucleus, means I cannot go out of this studio. What I will do, I will call office boy. Office boy will come to me. I will ask that office boy to bring some book. He will say, sir, please tell me the information which book I need to bring up. So I'll take a pen and paper. I write down the 10 books. That poor office boy do not know what kind of the book are they and what is use of them and all that. He is not aware. He just taken the information which I passed the information. So I am DNA. I have passed my information to that office boy, and office boy is RNA, messenger RNA. He will take my message to whom? To at the bookstall. right? Now DNA have passed its information to that messenger RNA. That poor guy that go out of this nucleus, means out of this office, out of nucleus, it goes out from this office as well. So outer side there is a protoplasm. The same way mRNA went to at the protoplasm. Now this messenger RNA, that office boy will go to a bookshop. That bookshop is ribosome. The name of bookshop is ribosome. It went to ribosome, means that bookshop, and saw that list of the 10 books. What is happening? Every time a bookseller is coming, right? That office boy are there in that bookshop. So every time that a boy is coming, read the name of the book, take out a book from the shelf and put the book and go out. Next boy is coming, reading the name of the book, and again putting the book according to the name and goes out. Like this way, the 10 boy came one by one. 10 boy came. They put a book on another book by one another book, and they go out. Right. The boys which are bringing the book, they were tRNA. Where the book was piled, that was the ribosome. When the bundle is made on the basis of my information, the bundle is released from there, and I got that bundle. So I was a DNA. This office was a nucleus. This door was a nuclear pore. That office boy was mRNA. That bookshop was ribosome. Those which are bringing the books, they were tRNA. Every book was amino acid. The bundle of book is polypeptide. Got it? Got everything or not?
Yes, sir.
You got my story? Now come to the real picture.
So where does these amino acids come from? Like from where does the tRNA?
Actually, amino acid, they keep on floating in amino acid, nitrogen, everything keep on floating in cytoplasm. That's why cytoplasm is called, that is pool. That is nucleotide pool. That is amino acid pool. So everything is floating over there. From where does they came? You have add the protein. You have eaten the protein. Protein went to your mouth, from mouth to your stomach, to intestine. In intestine, the protein have been broken down into amino acid with the help of proteases. Pepsin, trypsin, they broke down the protein into amino acid.
Yes,
that
sir.
amino acid have been absorbed from your blood, by blood, from your intestine to blood. Blood had distributed this amino acid across the body. From the blood, it had this amino acid have been taken by lymph. This lymph have given this amino acid to the cell. That is the source of amino acid. Got it?
Yes, sir.
Okay. So how does the translation take place? Look, this messenger RNA came out. This one, mRNA, it came out. First codon will be starting codon AUG. At AUG, this tRNA will bring amino acid methionine. This is anticodon. Formation of the initiation unit taken place. This is larger subunit, smaller subunit, larger and smaller subunit joined. So this is the formation of initiation complex. After the formation of initiation complex, initiation will take place. What will happen? A tRNA will come, and look, inside the messenger RNA, what is happening? One more thing I want to tell you. Inside the messenger RNA, there are three small rooms: A site, P site, and E site. At the P site, this A site is amino acyl site. Here from here the tRNA will enter. P site, polypeptide site. Here the peptide bond formation will take place. From E site, this tRNA will exit. So what we'll do? Imagine there are three rooms: A, P, and E. Every time a boy is coming with like a pot at the head, that is amino acid. It went inside the A site, then P site. When it went to the P site, when it was about to exit, now the next one came. From A site, next one came. When the next one came, what happened? Before exiting, it had put its picture on that another one, right? So this A1, the person who was A, He came over here. When he came over here, this E, the guy who's in E, he put his amino acid on the head of A. Now when this guy will exit, it will put its amino acid on the head of another one. That's how the polypeptide chain will be formed, right? The same thing is happening over here. This tRNA is coming. Now when the next one will come, then this will move shift over here. When the next one will come, this will shift here. Before exiting, it will put amino acid over here. By this way, the formation of peptide bond will take place between amino acid and the polypeptide chain will be formed. This is called elongation. When the polypeptide chain will be formed, at the end, when there will be stop codon, there is no amino acid from the stop codon. Now the polypeptide chain formation will stop and the chain will be released with the help of release factor. And this is called the termination of the chain, right? And this complex will dissociate, means amino acid, this largest subunit will dissociate, tRNA will dissociate, and small subunit will dissociate, everything will dissociate. And that's how the formation of polypeptide chain takes place. Tell me guys, is it clear or not?
Yes, sir. Sir, so every mRNA will have one start codon and one stop codon.
Exactly. Every at the starting, every mRNA will have initiation codon, and initiation codon is fixed. That will be AUG, methionine. But stop codon can be any of three.
And sir, what will happen to the last tRNA that will be here in the P room?
Exactly. The last tRNA will be at the P room. The release factor will come and will release the polypeptide chain.
Okay, sir.
Betsy, Iram, tell me, is that 100% clear?
Yes, sir.
Now,
Yes,
last
sir.
thing, what is the difference between the polypeptide chain and protein? This is polypeptide chain. The folding of polypeptide chain will decide its characteristic, that either it is going to be structural protein or it will become an enzyme or hormone. And the folding of the polypeptide chain will be done by a protein that is called chaperone. So chaperone is the protein. After release of polypeptide chain, they fold the polypeptide chain, make a polypeptide chain into protein and decide its characteristic. Chaperone protein. Please remember chaperone protein. It have been asked, that's why I have incorporated over here. Chaperone. Chaperone is the protein that decide the folding of polypeptide. If you want, please note down. Chaperone is the protein that determine the folding of polypeptide chain and decide its characteristic. So almost we have done with this chapter. There are only few things which are left. Only lac operon and human genome project. Only DNA fingerprinting. 3 topics are left. Very small topic. And then next class, in the first 15 or 10 minutes, we'll do this and we'll do some questions as well. And I ask you to please go through with the previous year question paper, right? And